we're this week, which is week 12, the first week after our spring break, we're going to finish up Unit 7 viruses. I'm hoping you guys were able to catch a little bit of rest and relaxation over break. Um, so this week we'll have three new viral PowerPoints that we'll be going over. The first one is a short introduction to immune responses to viral infections. And then the second PowerPoint will be a brief introduction to diagnostic tests for viruses, focusing on nucleic acid tests and antibody tests. And then the last PowerPoint will be on just a short introduction to viral vaccines, and then that will finish the viral unit. We're going to move Unit 8 prions to next week, week 13. All right, so we just wanted to take this opportunity to go over some of the details that will maybe help us understand some of the challenges that are going on during this COVID-19 novel coronavirus pandemic. All right, folks, so what I would suggest to make studying most efficient is for you to download, maybe print out the study guides that go with each of the PowerPoints. And what I thought I would do, again, trying to make this most efficient for you all, is I'll first briefly go over the study guide questions. And then if that's enough for you, then that's good. I'll go over the study guide questions, discuss really short answers, and then I'll go over the PowerPoint slides to provide more information. So folks, what I have in my hand right now, I printed out the study guide that goes along with this viral immune responses, or let's see, officially I guess we're calling it immune responses to the novel coronavirus 19. And the shortened title is viral immune responses. So let me just go through it really quickly. You'll see on the study guide, folks, that following each question there are slide numbers, and the slide numbers correspond to the slides in this PowerPoint. So I'm not sure if I'm coordinated enough to flip to the correct slides. Let me see if I can do this or not. Okay, so the first the first question, you guys, is what is the mucociliary escalator? How does it protect us against viral infections? And it's telling us to look at slide 3 through 5. So let me see if I can't get down here to, this is our slide 4. So the mucociliary escalator is a combination of ciliated cells, and we're going to focus on the upper respiratory tract, for example, the trachea. So within the trachea, we have these beautiful ciliated cells, and the cilia are these hair-like extensions, and they beat together. And they beat so that they can move, it's hard to see here, this mucus blanket. The cilia beat so they can move this mucus blanket up to the back of our throat, where either we can spit it out or we can swallow it. Now, the beauty of this mucociliary escalator is that all the garbage that we breathe in, every time we inhale, we're inhaling dust and dirt and microbes, right? They're going to get stuck in this microbial flypaper. That's what I think of as mucus blanket. And so, for example, folks, if we inhaled, say, a coronavirus, we'll just pretend that's one, right? It gets stuck in the mucus blanket. It won't be able to reach the epithelial cells. And then the mucus blanket will get, again, moved up to the back of our throat, where we can either spit it out, right? And the virus hopefully will die in the environment, or we could swallow it, and the virus would be killed in the acidic environment of our stomach. I think stomach pH is around 2, right? So this is a really, really important part of our normal defenses. So this next slide, I think, is just a little bit more detailed. Right, so this is pirated, guys. I'm pirating a lot of these slides. So showing you the histology of the mucociliary escalator. So here are these beautiful ciliated cells. This is a diagram showing the ciliated cells. These are I think they're called goblet cells that actually secrete the mucus, and here's that sticky blanket right there. Right, so that's a real important, like, first line of defenses, non-specific defenses against all kinds of respiratory pathogens. The, the, the problem is if folks say have been smoking for a long time, there's all kinds of toxins in cigarette smoke that can damage these ciliated cells. So people that have been smoking for long periods of time, their mucociliary escalator doesn't work very well. So we would guess they would be at higher risk for serious coronavirus infections. Furthermore, folks that have occupations where they're exposed to smoke, so think of firefighters, um, that's that's really kind of a high risk profession, right? Because they're going to be exposed to large amounts of smoke that can damage this. So as a consequence to damage of the mucociliary escalator, if we go back here, right, if the ciliated cells are damaged, they can't move that microbial flypaper to the back of the throat. So just by gravity then, that mucus can move downwards, carrying all the microbes that are stuck there, right? The microbes then can move down into the lower respiratory tract. And if they cause infection here, right, then the damage is going to reduce the ability of our bodies to absorb oxygen, right? Because that's kind of the purpose down here is we inhale oxygen-rich air, we absorb the oxygen into the bloodstream where the red blood cells pick it up, right? But if we have microbes, if we have, for example, coronavirus growing down here, that's going to cause a lot of damage and decrease our ability to absorb oxygen. So what I've noticed is one of the things they'll do if they think somebody maybe is a suspect coronavirus victim, they'll look for oxygen saturation, right? And if your oxygen saturation of your blood is down, that suggests something's wrong with your lungs, right? So that would be another piece of evidence suggesting you might have some kind of pneumonia going on. Okay. And then, folks, it says that next study guide question is which cells will kill our virus infected cells? See slide 6. So let's go down here, you guys. So this is getting into some of the immunology. Okay, so I know this is a really busy slide, you guys, but for the study guide 2, it says which cells below will kill our virus infected cells. So the answer, you guys, would be cells of the immune system. NK cells are natural killer cells. And then this is a second cell of the immune system, cytotoxic T lymphocytes. These cells are able to recognize our cells that have been infected by viruses. And their job is to kill our cells that have been infected by viruses. And you might think, gosh, why are they doing that? Well, remember, the viruses are obligate intracellular parasites. So if our immune system can quickly identify the cells, our cells that have been infected, and quickly kill them, the virus won't have a chance to replicate. So even though you are hurting some of your own cells, hopefully if the response is fast and strong enough, you'll stop viral replication. But this, you guys, is really important for us to understand that in infectious diseases where we're being infected by microbial pathogens, sometimes a lot of the damage is done by our immune response to the pathogen, right? So you'll hear me repeat over and over, our immune system is kind of like a double-edged sword, right? We absolutely need it, right? We need it to defend ourselves against the microbes, right? So one edge of the sword is killing the invading microbes, but then I think of it, you can get this rebound effect where the sword kind of bounces off the microbe, and the other side, which is also sharp, comes back and hurts you. So we can't live without our immune system, but our immune system can actually contribute to damage done, especially with viruses, right? And you know, I was just this whole debate, you guys, on the hydroxychloroquine, like the malarial drug, right? That some people have been touting could be used to treat the novel coronavirus infections. You know, this has really been puzzling me. But in the pharmacology books, it does say that it is used to control these immune disorder problems like lupus. These are like inappropriate immune responses, sometimes autoimmune responses, and so the hydroxychloroquine is used to help control symptoms related to lupus, which is an immune-mediated disease. And even though I didn't get it into this PowerPoint, you guys, maybe you saw some of the links where they think some of the people that are dying from COVID-19, they're dying from what's called a cytokine storm. And cytokines are chemical messengers that our cells release as part of the immune response. Now, the way it's supposed to work is you just have a little bit of cytokines released locally, right, where the pathogen is invading. But what happens in cytokine storms, our immune cells are triggered to release, you know, just a flood of cytokines that then flow throughout our body. and will cause changes to the vascular system like vasodilation, capillaries get leaky, your blood pressure goes down, that decreases blood flow to your tissues and organs, right? And then on top of it, right, if you have a pneumonia, excuse me, the infection in the lower respiratory tract, you already have decreased oxygen absorption, right? So what ends up happening then is we're going to decrease the supply of oxygen to our tissues, to our organs, and they start to die, right? And we'll see later in the medical micro pathogenesis section, at least with bacteria that cause sepsis, part of these changes that occur from these cytokine storms, we can also trigger the clotting cascade. So you can get further blockage of blood vessels with little tiny blood clots. And then the irony is, and I don't think this is true with the novel coronavirus, but it is true with some bacterial pathogens triggering sepsis. Eventually, you can use up all your clotting factors and then you end up hemorrhaging, bleeding out. Okay, so again, folks, study guide question 2, which cells below will kill our virus-infected cells? So we would choose the NK natural killer cells and the cytotoxic T lymphocytes, also known as TCs. All right, then question 3, folks, is what are the consequences of cell killing by the cells named previously? Why may this, why may this cell killing increase risk for pneumonia? So we just want to go back up here, you guys. So remember how we said with smokers, when they damage their mucociliary escalator here, right? Well, the viruses that replicate in these ciliated cells, right, if they cause lysis or decrease function, or they kill these cells, right, that's going to kill these ciliated cells. So again, that mucus blanket can't get moved up. And then, you guys, if our immune cells are helping to kill the cells that the coronavirus has invaded, right, that's also going to decrease function of the ciliated cells. So you won't get that mucus blanket being delivered, moved, transported to the back of the throat, where we can spit out the mucus with the trapped pathogens in it. Okay, all right, so let's see here. We'll just keep going. Okay, so again, folks, this is what I've been reading is when folks have these really serious lower respiratory tract infections with the coronavirus, it gives this ground glass appearance to chest radiographs, right, showing the damage and the results of all the damage that the coronavirus is causing. And again, the problem here is you have decreased oxygen absorption, right, when we have these pneumonias going on, and obviously that can kill us, right? We need oxygen to keep aerobic respiration occurring in our cells, and without aerobic respiration, our cells are going to start to die. All right, so now this is slide 9, and let's see where we are here, you guys. All right, so so for study guide question 4, it asks what are the 2 arms of specific adaptive immunity, right? So we're kind of jumping ahead of ourselves, you guys, but I think this is all right because we're actually giving you an introduction to immunology, which is the last unit of the course. So you'll recall we said the mucociliary escalator is one of our frontline defenses against not only coronavirus but all respiratory pathogens. So our mucous membranes and skin are described as nonspecific, natural or innate defenses, right? These are the defenses that are always on, they're always active, and they protect us against a wide range of pathogens. That's why they're called nonspecific. So skin mucous membranes are part of the innate nonspecific surface defenses, right? And then later we'll see that we have nonspecific innate interior defenses, but we will leave that till a little bit later. So if nonspecific defenses are kind of the first responders, right, trying to protect us, do we have second responders, right? Do we have a backup system in case our nonspecific defenses can't protect us against a pathogen? And the answer is yes. Really, the big guns of our immune system belong to a branch of the immune system called, and over the years, you guys, it's had so many different names, so I'll try to give them all here. The big guns of the immune system are the specific acquired adaptive immune responses. So acquired because they aren't always on. We first have to get exposed to the pathogen or the toxin or maybe a vaccine. Adaptive because this arm of the immune system lets us adapt to the pathogens that we encounter in our particular environment. Immunity means protection. Specific because these weapons are going to respond to very specific pathogens or toxins, right? They're not going to be active against a wide range of pathogens. And we'll see you guys, probably the biggest disadvantage of specific acquired adaptive immunity is it takes a while, 10 to 14 days, for them to be activated. So in 2 weeks you might die, right, from the coronavirus. So that's why we need those non-specific defenses. If the non-specific defenses can't get rid of the pathogen, at least they can slow it down so that we can activate these more powerful guns. I think of it, you guys, this is silly, but like in World War I, they had these huge cannon, huge, gigantic cannon that could level a city, but it took a long time to transport them, right, to the place that the cannon was going to be used. So that's what I think of these big guns, specific acquired adaptive immune responses, like these huge cannon, and it takes a long time to get them activated, right, to get them working. So, folks, there within the acquired adaptive immune response, there's two further branches: humoral immunity. Humoral meaning fluids, and fluids contain antibodies or immunoglobulins. And this is what we'll focus on the next few slides: is humoral or antibody immunoglobulin protection. We had already mentioned an example of the other branch of adaptive immunity, cell-mediated immunity. Remember how we talked about the cytotoxic T lymphocytes, how they're going to detect and destroy our virus-infected cells? They're part of what's called cell-mediated immunity, which isn't involving antibodies. Okay, so again, folks, going back to our question, it says, what are the two arms of specific adaptive immunity? And the answer would be humoral immunity, immunity provided by antibodies, and then the other branch is cell-mediated immunity, protection provided by cells, for example, cytotoxic T lymphocytes. The next study guide question, you guys, it asks, antibodies, also known as immunoglobulins, are made by which cells? And it says to go to slide 10. Right. Okay. So you guys, there's specific cells, white blood cells called B lymphocytes. Okay, and I should have. Oh, here we go. B lymphocytes. Right. We've already mentioned T lymphocytes. T stands for thymus. Right. B stands for bursa, or in humans we could say bone marrow. So this is a different type of white blood cell, a different type of lymphocyte. The B lymphocytes, sometimes called B cells, and the B cells, when they get triggered by invasion of a pathogen or exposure to a specific toxin, for example, or exposure to a vaccine, the B lymphocytes are going to start pumping out antibodies or immunoglobulins. And a B lymphocyte that's making antibodies is referred to as a plasma cell. But on our study guide, you guys, and on the exam, if you just say B lymphocyte or B cells, you'll be fine. And what we want to remember is that when B lymphocytes are first triggered to start making antibodies against what we call an antigen, an antigen is something that triggers a specific immune response. The problem is, again, guys, the antibody production is slow. It's going to take 10 to 14 days before we get good levels of antibodies. Another disadvantage of what we call this first time that we activate adaptive immunity, called the primary immune response. So it's going to be slow. It's going to be low, meaning we're not going to make a lot of antibodies. It won't last that long. And when we actually get into immunology, folks, we'll describe how the class of antibodies that's made, it isn't the best class, right? We're going to discover that the first antibodies made are called IgM, and then the second type of antibodies that will be made are called IgG. And IgG have the greatest variety of functions. Okay, so again, folks, to answer question, study guide 5, antibody immunoglobulins are made by which cells below? So the choice would be B lymphocytes or B cells. And then on question 6, folks, it's asking us, describe 2 ways antibodies can protect us from serious viral infections. So that's slide 11 and 12. So let's take a look here. And folks, this is just some of the functions of antibodies. Again, in the final unit, we'll go into more different functions of antibodies, but in this PowerPoint, we'll just talk about 2. So we've already talked about how antibodies can prevent attachment, say, of a virus or a toxin or a bacterium. And the antibodies do this by binding the adhesins on the surface of the pathogen or the equivalent on a toxin. And we said the fancy term for blocking attachment is neutralization. So one way that antibodies can protect us is by blocking attachment of the pathogen. And so these antibodies are called neutralizing antibodies. And we know definitely with viruses, if they can't attach to our cells, then they can't invade our cells and thus they can't replicate. So neutralizing antibodies, what a brilliant way to stop viral infection. You stop the first step, right? You block their ability to bind to our cells and therefore they can't replicate. And then folks, on slide 12, I don't think we've talked about this function of antibodies, and this function is called opsonization. So let's say this is some invading pathogen right here, right? And ideally what we'd like is when the pathogen first invades us, we'd like our phagocytic cells like macrophages or neutrophils to quickly attach to the pathogen, ingest it in the process of phagocytosis and kill it. Well, it turns out that our phagocytic cells have to have um, they have to have adhesins that bind to specific molecules on the surface of the pathogen. And if they don't have, maybe adhesins isn't the right term, maybe receptors, yeah. So this is a better term, you guys. The phagocytic cells have to have receptors so they can bind to specific molecules on the surface of the pathogen. And it's a big problem if our phagocytes lack receptors for the molecules on the surface of a pathogen, then our phagocytes can't attach to them. So this is where these opsonizing or opsonic antibodies come in. If the antibodies can first bind to the invading pathogen, coating the invading pathogen, or the fancy term is opsonizing the invading pathogen, then our phagocytes have receptors that can bind to those antibodies, right? So the little green dots here belong to the phagocyte. These are, we'll learn later, the fancy term are Fc receptors. The Fc is just this tip of this Y-shaped antibody, right? So if the antibodies first coat the pathogen, opsonize it, then the phagocyte can bind to the tips of the antibodies by these Fc receptors. And that's going to greatly increase phagocytosis and greatly increase killing of the invading pathogen, right? So this is another way that antibodies could help protect us against viral infections by opsonizing the viral pathogen, increasing phagocytic killing. Then study guide, guys. Study guide 7 is what is an important disadvantage of the primary immune response. And you'll recall, folks, that we said the primary immune response is the first time, for example, B lymphocytes or T lymphocytes encounter the pathogen. Okay, so in specific acquired immunity, we want to remember that there's this lag period of 10 to 14 days, right? So the first time those cells encounter a pathogen or an antigen or a toxin. So we say the problem with a primary immune response is it's slow, right? It takes a long, long time to get it operating, like, again, 10 to 14 days. And so we might argue, well, is there a way where we could speed up um, this arm of the immune system, right? This specific acquired immunity. And there's two ways we can do it if we're going to focus just on antibodies, right? So one way we can do it is through vaccination, right? And another way we can do it is through a process called passive immunity. This is when we give a patient antibodies made in another person. So let's just take a look at this, you guys. So again, going back here, if we're going to describe two ways to speed up immunity involving antibodies, we could talk about vaccination or we could talk about passive immunotherapy. So now, folks, question 9 says vaccination triggers production of blank cells, which later can trigger a faster, stronger, more effective secondary immune response. So this is slide 16. So let's flip forward here, you guys. Oops. Okay. So, oh, sorry. Before we go to slide 16, you guys, this is kind of a classic graph that we show when we're talking about immunology. So this is a graph where we're looking at antibody levels in blood against time, right? And this part of the graph is showing what happens during the primary immune response, the first time our B lymphocytes encounter an antigen, something that's going to trigger an adaptive immune response. And so here's like day 1 when we're first exposed to the pathogen. Here's 2 weeks later, day 14. So again, folks, you can see it takes almost 14 days, or probably here more like 10 days, to get an increase in antibody production. But notice again, you guys, it's slow, right? It's low, right? It doesn't last very long. And we'll see later again, you guys, it's going to be primarily an IgM response. And what we want is this IgG antibody here. But what this graph isn't showing us is the huge thing happening in the primary immune response is that our body is making these fantastic memory cells, memory B cells and memory T cells. And the job of the memory cells is to live long and literally remember that pathogen, remember that antigen, remember that toxin, so that if subsequently we're infected by the same pathogen or we're exposed to the same toxin, then those memory cells, it's almost like they're pre-activated, right? Those memory cells quickly become activated, right? So they're going to quickly start making antibodies, right? So it's going to be a faster response. Notice, you guys, how much more antibody we're making. So it's a stronger response. It's going to last longer, right? The secondary immune response, again, you guys, it's that better antibody IgG, primarily an IgG response. And what's awesome, you guys, we're going to make more memory cells here, right? More memory cells. So we can take advantage of our understanding of how the primary immune response will trigger production of those protective memory cells by purposely exposing ourselves to antigens of pathogens that won't harm us, right? And that's the whole idea of vaccination. We're artificially going to expose our immune system with substances from pathogens, sometimes even weakened pathogens themselves, hopefully weakened pathogens, attenuated pathogens that won't harm us, so that will trigger this primary immune response, right, with something that won't harm us, right, and make all those memory cells. So then subsequently, if the actual pathogen infects us, we'll have this stronger, faster, longer lasting, more effective, primarily IgG immune response, right? And so vaccination hopefully will protect us from serious harm, right? We might still get infected, right? But the pathogen hopefully will quickly be defeated by this stronger, longer lasting, more effective, what we call secondary immune response. And that's the whole idea behind vaccination. So question 9, folks, it says vaccination triggers production of, we're going to say memory cells, right? These memory cells are what will protect us. Memory cells give us lifelong immunity. Memory cells are the basis of vaccination, right? The memory cells can literally save our lives. And then let's see here, folks. What does it say here? So question 10. Let's see, let's move up here. Yeah, right. We really need, as you guys know, I don't need to tell you that, we really need a COVID vaccine, right? Hopefully within a year. So hopefully this time next year, you guys, this will be just kind of a bad memory for everybody. Okay, folks, so then the final question is, this is question 10. How many plasma, the liquid portion of blood from a person previously infected with COVID-19 who has recovered and is no longer infected, how may their plasma antibodies help a person currently infected with COVID-19? What type of immunity is this called? So you guys, this is the other way that we can speed up immunity. And it's actually not speeding up immunity, it's transferring antibodies from a person that has survived this infectious disease. We transfer antibodies in the liquid portion of their blood from their plasma. We transfer those antibodies to a person who's presently infected, right? So this is why people are, again, you guys, we so need that antibody test, right? So we can identify who's been infected, who is recovered, who could be antibody donors, right? We so need that. So this type of immunity is called passive immunity because we are not. Let me see. Let's say I'm infected, you guys, with the coronavirus, and I receive antibodies from one of you, right, that's had it and survived. So those antibodies will protect me just as we've described, but I didn't make them myself. And so instead of being called active immunity, it's called passive immunity, right? And so we would say this is a potentially another way to treat people, not just only with drugs, but with antibodies. So we could call this immunotherapy, right? So again, this is just going through it, you guys. So antibodies made by one person who's recovered can be transferred to another person who's suffering presently with the coronavirus. It's called passive immunity because we're not making the antibodies ourselves, we're receiving them from another person. But just again, you guys, just to emphasize, those passively acquired antibodies could neutralize the coronavirus, could opsonize the coronavirus, and that might help that person who's infected to survive, right? Because remember, their primary immune response is going to take up to 2 weeks to get activated, right? And they could die in that time. So again, folks, and I know you all know this, but we just so need um, we so need those tests that can identify who of us has been infected, who of us have antibodies, and one reason is so we can act as antibody donors, right, to people that are infected now. And secondly, what's going to be really important is to identify those of us who have been infected and have survived. We should have at least some good protection against being reinfected with the coronavirus. And that means, maybe like I was thinking I could volunteer to go in and clean hospital, rooms, right? You guys, a lot of you are on the front line. So our medical staff are just being overwhelmed. And I thought, if I knew I'd already had it, I could go help. Like I said, I could clean toilets, I could clean patients' rooms, right? So anyway, so folks, the next PowerPoint coming up is just a short introduction to diagnostic tests used for viruses, and this is going to be diagnostic tests that involves nucleic acids and antibodies. So we'll talk more about how we need these antibody based diagnostic tests. Okay, guys, so we'll close that there. And again, we'll have two more PowerPoints. The next one will be on diagnostic tests for viruses, and then the last one will be on viral vaccines, which hopefully you guys, it's good news. We have some really good vaccines that protect us against some really serious viral pathogens. And I know we can do this for the COVID. We're going to get a vaccine here.